In the last 2 videos, I showed you that anxiety isn't broken hardware, it's outdated software. And I gave you the exact protocol I used to permanently eliminate phobias through mental simulation. But I promised you something else. The neuroscience of why this actually works. Because understanding the mechanism isn't just intellectually satisfying, it's strategically necessary. When you know how your brain updates itself, you can troubleshoot when the protocol doesn't work the first time. You can calibrate intensity. You can predict what will and won't create change. So today we're going deep: memory reconsolidation, predictive processing, and the specific brain structures that make self-directed neuroplasticity possible. This is the science behind why you can deliberately reprogram your own nervous system. First, you need to understand what your brain is actually doing moment to moment. Your brain isn't passively receiving information from the world. It's constantly generating predictions about what's going to happen next and then checking those predictions against incoming sensory data. This framework is called predictive processing or predictive coding, developed extensively by neuroscientist Friston through his free energy principle, and elaborated by researchers like Andy Clark and Jacob Hoey. Neuroscientist Lisa Feldman Barrett extends this in her theory of constructed emotion. Your brain generates your emotional experience based on predictions derived from past experience, not simply as reactions to current stimuli. Here's how it works. Every experience you've ever had gets encoded as a probability distribution, what Bayesian statisticians call a prior. Your brain is running a continuous calculation. Given my past experience, what's most likely to happen next? When you walk into a social situation, your brain isn't neutrally observing. It's predicting. Based on past data, what's the probability of rejection? What's the probability of judgment? Can I handle the emotional intensity if things go badly? And here's the critical part. Your brain generates your emotional and physiological state based on these predictions before anything actually happens. This is why you can feel anxious walking into a room where nothing bad has occurred yet. Your brain predicted danger based on past data, and it's preparing your body accordingly. Anxiety isn't a response to the present. It's a response to your brain's prediction of the future based on past data. And that past data, that's where we can intervene. Now we get to the core mechanism that makes the protocol work: memory reconsolidation. For decades, neuroscientists believed that once a memory was consolidated, fully formed and stored, it was permanent, fixed. You could create new memories, but you couldn't edit old ones. That was wrong. In 2000, neuroscientist Nader and colleagues at NYU, working with LeDoux, published a landmark study in Nature that fundamentally changed our understanding of memory. They demonstrated that when a consolidated fear memory is recalled, it becomes temporarily unstable and susceptible to modification. This discovery revealed something extraordinary. Every time you recall a memory, it enters what's called a label state, a brief window where the memory trace can be modified before it gets restored. This process is called memory reconsolidation, and it's the biological mechanism that makes permanent change possible. Since that initial discovery, hundreds of studies have confirmed this mechanism across species and memory types. Researchers like Daniela Schiller at Mount Sinai have demonstrated its application to human fear memories. Here's the sequence. Step 1, retrieval. You activate the fear memory by thinking about or experiencing the trigger. Step 2, destabilization. The memory becomes neurochemically unstable for approximately 5 hours. This window varies but is well documented in the reconsolidation literature. Step 3, new learning. If you introduce contradictory experiential data during this window, it gets incorporated into the memory trace. Step 4, reconsolidation. The memory is restored, but now it contains the new information. This is why the mental simulation protocol works. When you vividly imagine dominating the threat, you're not creating a separate good memory alongside the bad memory. You're editing the original fear memory itself. Your brain can't easily distinguish between intensely imagined and real experiences. Both activate the same neural circuits, both trigger the same neurochemical cascades. Neuroscientist Giacomo Rizzolatti's work on mirror neurons and motor simulation supports this. The brain uses similar neural machinery for imagined and actual experiences. So when you repeatedly simulate dominance while the fear memory is active, you're installing contradictory data into the memory trace itself. After enough repetitions, the fear memory no longer codes for danger I can't handle. It codes for threat I can dominate. The memory didn't disappear, it was rewritten. Now let's connect this to the prediction framework. Your brain's predictions are based on its internal model of the world. That model was built from past experiences. And one of the most powerful ways your brain updates its model is through prediction errors, moments when reality contradicts what the brain expected. In computational neuroscience, this is formalized through Bayesian inference. Your brain maintains probability distributions called priors and updates them when new evidence, sensory data, contradicts predictions. Every time your brain predicts X and experiences Y instead, it has to update its probability distributions. Small prediction errors equal small updates. Large prediction errors equal large updates. This is formalized in Rescorla-Wagner learning theory and expanded in contemporary predictive coding frameworks. Here's why emotional intensity matters in the protocol. When you create a vivid, emotionally charged mental simulation where you dominate a threat, you're creating a massive prediction error. Your brain predicted helplessness and fear. Instead, it experienced power and aggression. The bigger the prediction error, the faster the model updates. This is why I said the simulations need to be absurdly over the top. You're not aiming for realistic. You're aiming for maximum contradiction to the existing prediction. And here's what happens over 50 to 100 repetitions. Each simulation is a data point. Your brain is Bayesian. It updates based on accumulated evidence. After enough contradictory data points, the probability distribution shifts. Old model: social situations equal 80% chance of rejection I can't handle. New model: social situations equal 20% chance of temporary discomfort I can easily handle. The prediction changes. The emotional response changes. The physiology changes. This is why it's permanent. You've fundamentally altered the predictive model that generates your anxiety in the first place. Let's get specific about what's happening where. The amygdala, your threat detection system. Joseph LeDoux's decades of research at NYU established the amygdala as the brain's primary threat detection and fear response center. His work identified two distinct fear pathways, what I've been calling the high road and low road. The low road, sensory thalamus to amygdala. Fast, automatic, bypasses conscious awareness. The high road, sensory thalamus to cortex to amygdala. Slower, involves conscious processing. In people with anxiety, the amygdala is hyperactive, oversensitized to potential threats. Research by Elizabeth Phelps at NYU has documented this hyperactivity in anxiety disorders using fMRI. When you run the mental simulations, you're doing two things. One, extinction learning. Repeatedly activating the amygdala without actual harm occurring teaches it to reduce its response. This is the mechanism behind exposure therapy, as documented by Michelle Krask at UCLA. 2. Competing activation. Dominance and aggression imagery activates circuits that inhibit fear circuits. Research on approach avoidance motivation shows these systems are neurologically incompatible. You cannot simultaneously activate withdrawal, fear, and approach aggression systems at full strength. The hippocampus, your memory context system. The hippocampus is responsible for contextualizing memories, encoding when and where something happened, whether it's past or present. Research by J. Douglas Bremner and others has shown that trauma and chronic anxiety cause measurable hippocampal atrophy and functional impairment. This is why traumatic memories feel like they're happening now instead of being clearly marked as past. Memory reconsolidation requires hippocampal involvement. When you update a fear memory through the protocol, you're restoring proper temporal tagging through a process called pattern separation, the hippocampus's ability to distinguish similar experiences and update stored representations. The prefrontal cortex, your executive control center. The medial prefrontal cortex, or MPFC, particularly the ventromedial prefrontal cortex, regulates emotional responses through top-down control. Research by Kevin Oxner at Columbia has documented how the MPFC can inhibit amygdala activation during emotion regulation. But here's the problem. In anxiety, this prefrontal control is weakened. the amygdala overpowers it. Helen Mayberg's work on depression and anxiety shows reduced MPFC activity and connectivity. When you run mental simulations, you're strengthening prefrontal to amygdala connections. Each time you consciously direct your imagination toward dominance, you're building the neural pathways that allow your prefrontal cortex to regulate limbic responses. Over time, this gives you genuine top-down control, not through suppression, but through structural strengthening. The insula, your interoceptive awareness system. The insula processes internal body sensations and generates your emotional experience from physiological states. D. Bud Craig's research established the insula as the primary interoceptive cortex. Lisa Feldman Barrett's work demonstrates that emotions are constructed from interoceptive sensations combined with conceptual predictions. The insula interprets body signals: racing heart, tight chest, shallow breathing. In anxiety, the insula misinterprets normal body sensations as danger signals. Racing heart equals panic attack coming. Tight chest equals something's wrong. Mental simulations work on the insula because you're deliberately pairing body sensations, elevated heart rate, tension with dominance instead of fear. You're reconditioning the interpretive framework, what Barrett calls recategorization. After enough repetitions, the same physiological arousal that used to mean danger now means activation or even readiness. You're not eliminating the sensation. You're changing what it signifies. Now you understand why talk therapy alone frequently doesn't work for anxiety. Talking about the fear activates the high road, the cortical conscious processing pathway. But anxiety lives in the low road, the amygdala-driven, body-based, preconscious system that LeDoux identified. You can understand your anxiety perfectly and still have it because understanding happens in the wrong brain region. Cognitive behavioral therapy, or CBT, tries to bridge this by creating real-world exposure. When it works through the same mechanisms I've described: prediction errors and memory reconsolidation. Michelle Krask's inhibitory learning model explicitly leverages prediction error to update fear memories. But real-world exposure has limitations. 1. It's often too threatening to attempt. 2. It's logistically difficult to engineer repeated exposures. 3. The fear response can be so overwhelming that it reinforces the fear instead of updating it. What's called sensitization. Mental simulation bypasses these limitations. 1. You control the intensity. You can start at a level your system can handle. 2. You can run 10 simulations in 20 minutes from your bedroom. 3. You maintain enough conscious control to ensure dominance rather than overwhelm. You're hacking the same neuroplastic mechanisms that researchers like Nader, LeDoux, Krask, and Barrett have documented, but with precision control. Based on the neuroscience, here are the variables that determine success. 1. Emotional intensity. Memory reconsolidation requires emotional arousal. The neurochemical cascade that makes memories label, involving norepinephrine and stress hormones, only occurs when the emotion system is activated. This is documented in Nader's original work and subsequent studies. If your simulations don't trigger physical sensations, elevated heart rate, muscle tension, heat, they're not intense enough to cause reconsolidation. 2. Repetition frequency. Neural pathways strengthen through repeated activation. You need enough repetitions for the new pathway to become the dominant pathway. This is Hebbian plasticity, named after psychologist Donald Hebb, who established the principle neurons that fire together, wire together in 1949. Modern neuroscience has confirmed this at the synaptic level through long-term potentiation, or LTP research. 3. Experiential contradiction. The new data has to actually contradict the old prediction. Imagining yourself coping with anxiety isn't enough. That's consistent with the prediction that there's something to cope with. You need to imagine eliminating the threat entirely through dominance. That's the data that creates the prediction error large enough to rewrite the fear memory. For body involvement, the simulation has to be embodied. You need to feel it in your body, not just visualize it abstractly. This activates the insula and creates genuine interoceptive learning, as documented in Craig's and Barrett's research. Your body needs to experience the shift from fear state to dominant state. This is why I can say with confidence that the protocol doesn't just manage anxiety, it eliminates it. You're not applying a coping skill. You're rewriting the source code. Karim Nader's discovery of memory reconsolidation allows you to edit the fear memories directly. Carl Friston's and Lisa Feldman Barrett's work on prediction error learning allows you to update your brain's threat assessment model. The specific brain structures involved, amygdala, hippocampus, prefrontal cortex, insula, are all plastic as documented across decades of neuroscience research. They change based on experience. And mental simulation, supported by Rizzolotti's work on motor simulation and mirror neurons, provides the experiential data these structures need to update. You are running a neuroscience experiment on yourself. And when you understand the mechanisms this precisely, you can troubleshoot, adjust, and optimize until you get the result you want. Next video, troubleshooting the protocol. What to do when it doesn't work the first time, how to calibrate intensity, and how to identify whether you're targeting the right fear pattern. But for now, you have the science. You have the protocol. Run the experiment.